So, my dear friends, we have again joined here to discuss something related to the burning topic, the COVID. Today, the topic we have chosen is to look at the vaccines related to COVID. So, I'm going to give you an update till what has happened today morning. So, as usual, we use all the international resources, and this is a vaccine landscape. And the presentation is available on the website and also on the YouTube. It will be available after the talk on the YouTube also. Please make a note of it. So, sincere thanks and acknowledgement has to be submitted to all those people who have contributed to the science. As always, nothing is mine. Idam na mama means that we are not owning any sort of ownership on any of the data that is presented. What is presented here is only a collated information, a systematically streamlined and scientifically presented. Purely for educative purpose, as a CME, I'm saying this because this will go on the YouTube and nobody need to find fault that we are using some of that material. No commercial interest whatsoever is involved in this. And thanks to all the international and national web resources, specifically, I have used quite a lot of material from Dr. Kushru Aristu's presentation on this topic, and also from clinicaloptions.com slide set on COVID, particularly on the vaccine area. And there is a YouTube link, beautiful YouTube link. All of you can also view this medcram.com, wherein a professor has explained the various vaccines. So an update on COVID vaccine, this is what we are going to look at. What is an urgent need for vaccine in this particular scenario in COVID? The disease is highly transmissible and very easily transmitted also. Okay? It causes morbidity and significant mortality also. We have seen our kith and kin, friends, neighbors dying. Secondary attack rates are quite high. If a person in the family gets it, 3 or 4 others are infected. That is secondary attack rate. Reinfection after COVID is being seen here and there, not very common, but still could be a problem in future. Another important thing is there is an asymptomatic illness in this disease. Means what? Patient will not have any symptoms, but still he will be harboring the virus and transmitting it. Okay? And there is no effective and specific antiviral agent to treat the disease as yet. Though we have 3 or 4 drugs which are on the borderline, nothing has crossed the boundary and established itself as a specific treatment. And the disease is novel. That's another thing. We don't have the herd immunity. We don't have any exposure to the virus earlier. So how do the body know? How does the body know that this virus? So there is no what is called the innate or herd immunity. Today morning, 7 a.m., this is the caseload. Just we have crossed 1 crore. And the deaths are 1.45. Many of our kith and kin are in this. Doctors also. The pandemic global cases are, you can see here, 75 million cases. We have 10 million out of that 75. And we have a global death rate of around 1.68 million or 16.8 lakh people died. No other disease in the memory of mankind who are living now has killed so many people. So definitely there is an urgent need for the vaccine. All of you know this SARS-CoV virus. We looked at it. This is the spike protein. There is also the envelope protein. There is the RNA inside. This is a single-stranded positive sense RNA. What is positive sense? I will tell you in a while. And then the spike glycoproteins are also there. And there are a nucleocapsid core is there. And the main important interest is the spike protein and the RNA core, to which we are going to see some vaccines. Traditionally, a vaccine development pathway is like this. So what will happen is there will be discovery and target validation of which particular area of the pathogen to be targeted. Then there will be a preclinical stage, and there will be what is called the translational medicine. So the lab will identify, we identify the antigen that comes into the sort of reckoning, and to which against the vaccine is prepared. manufacturing development, initial bioprocesses and formulation, analyticals and all that, and clinical assay of the antibody titers will take place. All this is in the lab. Nothing is in the human being. Then come the phase 1 trials, where around volunteers, 10 to 20 volunteers or 30 volunteers are given the vaccine to see how it works, that there are any side effects and things like that, not to look at the efficacy at this stage. Then comes what is called the phase 2 trials, where safety and immunogenicity, the ability to produce antibodies, is also studied over hundreds and 200, 300, 500 people. Then come the phase 3 trial, where safety, efficacy, regulatory approval, everything will be taken into consideration, and adverse drug reactions will be studied, and disease prevention will be studied, and the duration also will be longer. Here at least, minimum of 15,000 people are involved here, 1,000 plus people here. Then it goes to regulatory review, like the Director General of Drugs Control in India or the FDA in USA, they will approve and then allow the vaccine to come into this. So normally you see 3 to 8 years in the lab, 2 to 10 years in the phase trials, phase 1 to 3, and 1 to 2 years in the approval. Roughly around 20 years each vaccine was taking for us to get approved. If we were to wait 20 years, when are we going to conquer the COVID? So there must be a way to fast track those things. That's what exactly has been done. So the rapidity matches well with the speed, age, and also the requirement. Necessity is the mother of invention. Global pandemic emergency has prompted scientists to look all over the globe to find out a very quick solution to that, and also a durable one. First SARS-CoV genome sequencing has been done somewhere in around May, and within 6 months, all the phase 1 trials are over. And I think March they have done the sequencing. By September, phase 1 trials are over. Phase 2, phase 3 trials also are completed for some of the vaccines. The rapid development of COVID-19 vaccines is enabled because of the following factors. All of us might have a skepticism why they have brought the vaccine so fast. And the reason is we have a prior knowledge of the role of spike protein in the COVID pathogenesis, because the same spike protein was the damager in the similar infections we had earlier. So this is already known. Neutralizing antibodies against the spike protein is essential for protection. This also is already known. And the third thing is the evolution of nucleic acid vaccine technology platforms allows prompt manufacture of large number of doses in no time. What is that? The nucleic acid vaccine technology is where you use the RNA template to multiply the vaccine in the lab. It is just like a Xerox machine. Once you have the original there, just put it on that and then get copies any number you want. It's a question of time, how many doses you want, you can get it. Just about the time that is taken to make around 100 copies from one page, around 10 times that will be the lab replication time here. Many developmental activities are conducted parallelly and not sequentially. Sequentially means one after the other, it takes time. If you parallelly do certain things, then naturally the time is cut down. This is how we are able to get a quick vaccine into the market. Now, how many vaccines are there in total? You can see here, 87 vaccines are in the preclinical stage. In the lab stage, we said 38 are in the phase 1, 16 are in the phase 2, 12 are in the phase 3, and 2 are already approved by the European health authorities or the FDA. Two vaccines are already approved, and the third one is just knocking the door for approval. So we may have by tomorrow or day after, once the approval for the AstraZeneca vaccine comes, there will be 3 vaccines that are approved. So we now know somewhere around 140, 150 vaccines are being tried, of which 2 have come here. That means there is a lot going on here, and some of them might come down and then be ready for approval in months to come. Okay. Now, we have to contrast 2 important things. One is the infection, the other is the disease. Infection means the virus is replicating in the body. Patient may or may not have symptoms or disease, and he is shedding the virus and transmitting to other people. He is a risk to the community, and such a stage is known as the infection. Then the disease is patient gets the symptoms and it becomes symptomatic COVID disease, and then he may have milder, moderate, or severe disease, or may have complications. Why this is important? If we are using a vaccine, does it prevent the infection? Does it prevent the disease? That is the question. If it does both, it's very nice. If it only prevents disease and doesn't prevent infection, it cannot take care of the transmission. Okay, so for the reason of the transmission, we require a vaccine which can prevent infection and also prevent disease. Now, traditionally, you see, we know angiotensin converting enzyme 2 receptor is the receptor site where the spike protein gets attached and gets entry into the body. Okay, and there are several ways you can inactivate the spike protein by different types of antibodies here. These antibodies prevent the spike protein from interacting with the receptor and entry into the cell. Supposing there is no antibody is there, or patient is not able to generate these antibodies, or the time is too short for the antibodies to develop, then this directly goes and then gets into the system. It enters into the AC receptor and enters into the system. Now, what vaccine is trying to do is build up the antibodies here, so much so it cannot directly go and attack the ACE2 receptors, and then entry of the virus is denied or delayed, mostly denied. So how is the vaccine developed? There is a patient, let us say, who recovered from convalescent plasma, and that patient gives the plasma that is called the convalescent plasma. From that, we isolate the antibody, and in vitro, they do a functional assay in the lab about what is this antibody, what is its chemical nature, what is its structure, what is the amino acid sequencing and all. Then put it into the in vitro assay in the animals, particularly the mice and guinea pigs, and then do a functional assay about the antibodies generated by the mice against these things, and pick up those antibodies and see to which antigen they are attached, which is the antigen that is attracted, that the antibody is attracted to, and that is the antigen which we have to pick up. That antigen is isolated and that is preclinically evaluated in the phase, and then in the volunteers in the phase 1, and then the vaccine. All the story is there. So initially there will be some COVID patient who had recovered. There will be antibodies in them. Those antibodies are picked up in the lab. They are assayed, and then that is called in vitro. Then in the animal studies, they do in vivo. Then the antibodies are picked up, and the picked up antibodies are analyzed to find out which are the antigens to which the antibody is attached. And that is the antigen which has caused the disease here. And that antigen is injected into the animals to generate antibodies and see whether that is the same antibody as this and whether that is protecting. So this is the natural story for any disease. Now, we need to understand there is not one type of COVID vaccine, there are several types of COVID vaccines. So whenever we vaccinate, we are injecting the material, antigenic material, and stimulating the immunity in the body of the individuals. So that is called the active immunity. Passive immunity means we don't give the antigen, we straight away give the antibody, either a convalescent plasma or a monoclonal antibody or a polyclonal antibody or an mRNA-induced antibody. These are the various ways of passive immunity. So passive immunity, a lot of trials have been done on convalescent plasma, and the results are equivocal. Some are saying it is helping, some are saying it is not helping. Then we also had the bamlanivimab also is there, which is a monoclonal antibody specific to the COVID antigen. And there was a polyclonal antibody, which is called the Regeneron antibody, which has been touted as one of the very good effective antibodies. Both are effective. And also now we have a newer technology called the MIT, mRNA-induced antibody. Remember, this is not a vaccine. This is a passive immunity. Whereas when we come to antigens, this is vaccination, where you give the antigenic material into the body and allow the body to develop the antibodies against the antigen, and that is what is known as the active immunity. In the active immunity, we have DNA vaccines, Inovio, RNA vaccine, Moderna, and then Pfizer vaccine, viral vectors like Oxford-Zeneca, and then the CanSino Biologicals and Janssen's vaccine, and Gamaleya Sputnik Russian vaccine. These are all viral vectors. Viral subunits, Novavax, Adaptavac, Clover Biopharma, live attenuated, you can see the names here. Inactivated virus, again Sinovac, Sinopharma from China. VPL virus-like particles, split virus vaccines, example flu vaccines, and RNP ribonucleoprotein vaccines. So there are a host of ways of generating the antigen to be presented to the body to develop this one. So we need to understand what is the role and importance of each type of these approaches. Among them, the ones which are already approved are Moderna and then the Pfizer. It is called Moderna because mRNA, modern vaccine, it's called Moderna. Pfizer BioNTech vaccine also is an RNA vaccine. Moderna, Pfizer vaccine and AstraZeneca vaccine. This is also approved now. Johnson and Novavax are also not lagging very much behind. So let us see what is the importance of each type of vaccine and what are the advantages and disadvantages of a DNA versus an RNA or an mRNA vaccine versus viral vector vaccines. Now you see that basically it's a DNA virus or an DNA vaccine or an RNA vaccine or a protein-based vaccine. And the protein-based vaccines can be virus directly presented as inactivated or attenuated. There is a difference between attenuated and inactivated. When you present the DNA, there will be two types, vector replicating, viral non-replicating and replicating, and then the mRNA. So we have basically DNA type of vaccines, RNA type of vaccines, protein-based vaccines. In the DNA, vector replicating and vector non-replicating, and then inactivated and attenuated are here. Here we have the RNA tagged to the LNPs. So let us see, this is the total number of vaccines if they are there. Protein-based are 44. Viral vector non-replicating are 16. Viral vector replicating are 14. Others 10. DNA vaccines are 12. RNA vaccines are 20. RNA vaccines are 20 in number. Okay. SARS-CoV live attenuated only 3. Inactivated 8. This number will be very large in the previous infections. Say at 20 years, 30 years back, if you go, we have only live attenuated or inactivated vaccines. Today, we will not travel in the bullock cart. Will we ever travel from one place to another place in a bullock cart now? The speed has come. The modern science has allowed us to shift on to at least the protein-based or to the RNA-based or to the vector-based vaccines. So let us see each one of them. Each one of the type of those vaccines. We can't go through all those 140 vaccines, but we can see each type. So basically, here we are dividing the vaccines, active immunity vaccines into nucleic acid vaccines, protein vaccines, and viral vaccines. Viral vaccines means which are live attenuated or inactivated. Nucleic acids can be DNA, can be RNA, can be viral vector vaccines. 3 types. So under the nucleic acid vaccines, we have a DNA vaccine, we have an RNA group of vaccines, we have viral vector vaccines. Under the protein, viral subunit, split virus vaccine, virus-like particle vaccines. When you have the viral vaccines, directly use the coronavirus to be injected into the body, either you live attenuate it or you inactivate the coronavirus and give it. So these are the various vaccines that are being named there. And what is the company there, what is the mechanism, what is the current scenario. Now we know the ones in the red, Pfizer and Moderna, are the RNA vaccine, and AstraZeneca viral vector vaccine, and CanSino, Janssen and Gamaleya Russian Sputnik vaccine are readily available and almost ready to be used. These 2 are being already used. These are ready to be used. And Novavax also is catching behind. Others are down the line. Now, status of the vaccines. What type of the vaccine is that? How many doses are required? What is the route? And what are the phase 1 trials or phase 3 trials completed? Or what is the status of it? Now, see, most of them have completed phase 3. They are in the phase 3 trials, and 4 of them have completed the phase 3 trials. Only 2 are in the phase 2. They are intranasal and intradermal vaccines. Barring that, all of them are intramuscular injection. And all of them require 2 doses, except the CanSino, which is the Chinese vaccine. They're advocating 1 dose, the exclamation mark by the scientific community. Okay, one dose may not work. That's what everyone feels. We from the studies we know that a booster in 21 to 28 days is required for all the things. So generally, remember that most all the vaccines that are going to come up are intramuscular. All of them are either completed or in the phase 3. 4 of them, 2 being already used, the Moderna and BioNTech Pfizer, and this is already being used in Russia, and this is available and knocking the door for approval. And then the type of vaccines, these are the RNA vaccines, this is the viral vector non-replicating, this is also viral vector non-replicating. What does that mean? I will let you know. And mostly it is a 2 doses, 0 to 14, 0 21, 0 28. Minimum is 2 weeks interval, maximum is 4 weeks or maybe 3 weeks. The first dose, then 14 days or 21 days or 28 days as listed here by the investigators here. So in summary, we have 4 vaccines on the cards readily. 1, the Moderna, the BioNTech Pfizer, readily available, being used in Europe, European countries, Britain, and as well as in United States. Gemelli Sputnik being used in Russia, Oxford AstraZeneca. This is what India also is trying. Oxford AstraZeneca trials are going on in India. Anytime this may be available for immunization. If that is so, India would probably go in for Oxford AstraZeneca or maybe Moderna vaccine. We don't know what government will choose, or they may choose a sort of a cocktail of vaccines which they would like to use, different scenarios. Now, what about each one of these? Why multiple vaccines? Why do I require so many methods of this one? Why so many people say? Everyone wants to make a buck out of it. It's a lot of demand is there. Millions of doses are required. If a company's dose, if it can take even 10% of the share of the vaccine market, it's a lot of money. So everybody is putting their best effort to generate a vaccine. That is not a problem. So out of the 14 vaccines in clinical trial, 114 candidate vaccines in the preclinical evaluation, immune memory by the vaccine is what will protect the person against subsequent COVID infection. And multiple vaccines are always good because once there is competition, there will be a chance to get down the price and also to see that whichever is the optimum one in terms of side effects and durability of immunity, one can choose. Now, the phasing of the vaccine will be usually in our country also. This will be followed. Healthcare professionals, high-risk group people who are 65 and above will be the ones who will get at nursing home residents. And these are the vulnerable population. And those people with essential workers like police people and all the other essential workers and high-risk medical conditions, people who have comorbidities and people who are more than 65. Already Government of India is preparing lists, and anytime it will be phased into phase 1A and 1B and 2, and these will be the phases recommended by the CDC in United States. More or less similar pattern also will be followed in India in terms of prioritization of vaccine, because so India has got 130 crore population, and we have to have that many doses to immunize everyone. And everyone should accept the vaccine also. That's another important thing. So why 2 doses? This is another question. So this is when you have an initial infection or you vaccinate here, the immunity starts coming up and then goes down. For the immunity to be at a reasonably high level to protect, we require a second dose of the vaccine, which is called, we don't call it as a booster here. Booster is something which is given after 1 or 2 years. It is actually the schedule of the vaccine itself is 2 doses. But just like the hepatitis has got 3 doses, here we have 2 doses, 114 or 121 or 128. And the second dose further augments the antibody response, and that will remain for memory antibodies, and immune memory will be there both in the T cells and in the B cells. The primary response in the immune system is to generate the antibodies for the COVID virus, and that immune memory will be remembered by the memory T cells and memory B cells. Remember, in this particular disease, both the T cell immunity and the B cell immunity both are playing an important part. The cell-mediated immunity as well as the humoral antibodies, both are essential, and one has to develop both the things to ward off the future infections and damage. Now, what is the global picture? Which are the countries, different countries trying? Of course, India is trying basically the AstraZeneca Oxford vaccine and also CanSino vaccine also are being tried here. And you can see this if you enlarge the map in your PDF, which I'm going to circulate, you can see which country is trying which particular vaccine. And there are only very few countries which are not, there are many countries which are not making any trials. All the gray zones are, they're not doing any trials. The colored areas only are the ones which are doing the vaccine trials. Now, US has procured already this many doses of each particular vaccine. They have pre-ordered so that, the once it is approved, the population will get it. For example, India, you look at here, and this is, around 100 million doses. 200 is here, and 100 million is, 1 crore, 10 million. Something around that we have ordered now, and AstraZeneca vaccine is the one which we are going to probably use, and also probably Pfizer BioNTech also. 20 million doses we have asked. AstraZeneca we have asked 32, and different countries have already asked for different things. And if you look at the orange color, that is the Moderna vaccine, and this color is the other vaccines. Now, each vaccine to be explained very clearly for us. Now, types of vaccines we told, either they can be DNA, or they can be RNA, or they can be protein. DNA is the genetic information which resides in the nucleus. There won't be any DNA in the cytoplasm. This genetic information is transcribed. Transcription is nothing but original Aadhaar card and a Xerox copy of the Aadhaar card. You keep the original Aadhaar card in the chest, that is in the nucleus, and whenever the copy is required, just take a copy of that and give it to the bank. Whenever a protein is required, the body brings a copy of that, and that copy is called the RNA or the messenger RNA or mRNA. This mRNA is not in the nucleus, it is in the cytoplasm. And looking at the mRNA, the protein sequence is arranged and the protein is prepared. And the prepared protein is the antibody which will interact with the antigen of the virus. So this is the one which is going to do the job, and the blueprint for that in a Xerox copy is here. The master blueprint is in the chest in the nucleus. So all mRNA vaccines are going to introduce the mRNA into the cytoplasm and not into the nucleus. So much so if it is into the nucleus, mutations will be more, transmission to the offspring, and then so many other difficulties might occur if it is in the DNA. So why then the DNA vaccines? DNA vaccines are also being tried because once the DNA vaccines are used, they're making sure that the RNA only is released into the system, and that RNA or messenger RNA, mRNA, will produce the proteins, and these proteins will develop the immune response, the antibodies from the plasmacytes, and then they will interact with the antigen and then kill the virus and not allow it to infect our body. Okay, when we use an RNA, we have bypassed that transcription problem. This is called transcription or Xerox copy. So each time a blueprint is required, it has to go there and copy and come. And that problem is solved, and we are directly giving the RNA into the cytoplasm, not into the nucleus. And from this, the protein is produced, and that protein produces immune response. Whereas if you use proteins themselves, viral proteins themselves, to trigger the antibody response, you can do that, but the problem is the proteins are very short-lived, and they may be inactivated very fast before the immunity actually develops. That is the difficulty with this thing. SARS-CoV outbreak has promoted the rapid expansion of several technological platforms, including the DNA and RNA vaccines, never before clinically tested in human beings. This is the first time, if at all, in the human race will be using a DNA or an RNA vaccine. Earlier, we were all using protein, live attenuated, inactivated vaccine. Rather than producing a viral protein in the lab, deliver DNA and RNA direct our cells to make part of the viral protein that do not cause the disease. What are we going to do? Instead of giving the antibodies directly in the lab, we are showing the copy, the blueprint to the body and asking the body to manufacture the proteins. Today, rapid production of DNA and RNA in large amounts requires the sequence of the virus genetic material. Around 50 or 20 years back, nobody knows what is the genome and how to structure it. Now give me a piece of material, immediately the genome is available. Immediately the viral genome is known in another 2 months time after the virus has come out. Maybe around March time, we know the genome. From that genome, that is nothing but the protein sequence in the mRNA is the genome. And that RNA in the virus, the genome is picked up, and from the genome, the sequence of SARS-CoV virus is identified as early as January 2020. And within 6 months, we have all the phase 1 trials. As novel vaccines are being developed, like the protein subunit vaccine and split vaccines, production of virus-like particles to mRNAs making antibodies directly. So that is another story. Now, what are DNA vaccines? For the DNA vaccines, what you do is, we take a part of the viral DNA, the COVID, this is all COVID genome, let us say. This is all the total genetic information of the COVID virus. We know the S protein is important. Only cut that part of the S protein, and that S protein is incorporated into a plasmid. And with the plasmid, this is given into the body. Why the plasmid? The plasmid is to see that the body doesn't degrade the protein that is given. After all, this is a protein, and why a body may degrade the protein. So with the plasmid it is given, and then it goes into the dendritic cells or the macrophages there, and these macrophages pick up the abnormal protein, knowing that it is something foreign. They will eat it and engulf it, and they will present it to the immune system. This is called the antigen presenting cells, APCs. Once it goes to the immune system, the B cells and T cells, two lineages are there. They will say, oh, this is a foreign protein. So let us dissolve the capsid plasmid part and see the actual virus, actual viral protein. This is not the full virus at all. A small part of the spike protein genome. It's not even spike protein. Spike protein is the manifested form of the genome, and this is the genome part of the virus, which we are showing to the body. The memory cells transcribe immediately into mRNA. This mRNA is translated into a protein. That protein is assembled into host antibody. This is the mechanism here. So what are we doing? We are giving the genetic material, part of the genetic material of the virus, a small part of that, combined with the plasmid, injected into the body. Spike protein or S protein in the COVID, and the gene attached to a plasmid for delivery into the body. Plasmid is used so that the body does not deteriorate the foreign gene before it can produce immunity. Once administered, the DNAs are taken up by the host cells, by the dendritic cells, by the macrophages, by the antigen presenting cells, and they are transcribed into the Xerox copy or the mRNA. This mRNA is shown to the immune cells. Immune cells produce the S protein as per the sequence here, and that protein, spike protein, develops, the antibodies are developed, and these block the attachment of the virus to the ACE2 inhibitors by inactivating the viral spikes. That is the mechanism. What is the vaccine that is under investigation? Innovia Pharma USA is developing a DNA type of vaccine. Now, what is the mRNA vaccine? Here, we are not using the double-stranded DNA here. We are using double-stranded DNA. Instead, what we did was, we just picked up the mRNA sequence of the spike protein, because the moment I put it in the body by injection, the protein is destroyed. The mRNA is just destroyed by our body. To protect it from that, from the lab, what we do is, we encapsulate with a lipid layer or fat layer. This lipid layer is called the LNP, lipid nanoparticle, along with the mRNA inside. Once it is in the body, this LNP particle along with the RNA is taken by the macrophages, and lipases in the macrophage digest the lipid layer, expose the Xerox print there. From the file, it will open up, and this is presented to similarly to the antibody producing cells or the immunocytes there, the B cells and the T cells. Spike proteins, either antibody or T cell response is generated. This is the mechanism. What is the basic difference? There, the DNA material would have entered the nucleus. Here, the RNA material, DNA material is not used. RNA material only is used, doesn't enter the nucleus. It is only in the cytoplasm. And the two vaccines are Moderna and Pfizer BioNTech. These are the two mRNA vaccines. If you ask me, did we ever use mRNA vaccines or viral DNA vaccines? No. This is the first time human race is getting such a sort of experience. So, for example, first time when mobile phones are introduced, everybody was wondering what is the use of it. Now everyone uses it. So as technology is going, and WhatsApp was very difficult to use initially, but everyone knows how to use WhatsApp. Technology embracing is a question of time, but if it is advantageous and safer, it's better to use the technology. So the RNA and DNA vaccines strike a balance between generating an effective immune response and the ease of production. These generate a very good immune response and very easy to produce in the lab. That is the great advantage of using an RNA or a DNA vaccine, particularly an RNA vaccine. Same time, DNA RNA vaccines are cheaper to produce as the genetic material can be mass produced, just making Xerox copies of that. We are not producing proteins. Proteins are produced by the body. We are only just giving the print, how the protein has to be prepared by the body. That's why it is much cheaper. Okay, once DNA and RNA is taken up by the cell, cell can produce and show the protein on the cell surface to stimulate the immune response. They are also very safe to administer. This is one very great advantage. In immunocompromised, immunosuppressed, elderly people, these vaccines can be easily given, because if you use the protein vaccines, you are likely to trigger some reactions and disease in people of this category, whereas RNA and mRNA vaccines can be universally given without any reservation for people who are allergic, people who are immunosuppressed, people who are immunocompromised, people with no pathogenic or infectious components are injected. We are not injecting any part of the virus which is pathogenic. We are only taking the genetic material of the virus, which codes for the spike protein of the virus. Not even the spike protein we are using. So we are there very safe. All the same, since they are newer technology, there could be some long-term problems. Everybody is keeping their fingers crossed and waiting. But it appears that mRNA vaccines are very safe from the technology that we have. This mRNA technology is being used not for vaccines, but for drug development in the pharma industry, to develop large quantities of drug or antibodies. For example, the monoclonal antibodies against several diseases, we are developing using the mRNA technique only. First time we are using to develop it as a vaccine. Okay, what are vector vaccines? Vector is a carrier. Vector means a carrier. Here, what will happen is, this is the SARS-CoV virus, and this is the genome of the virus. This SARS-CoV outer coat is dissolved, and the genome, that is the RNA of that, is put into another virus, which is not pathogenic to human being. For example, adenovirus 5, adenovirus 27, these are not pathogenic to human beings. Into that adenovirus, you add this and inject this one. What will happen? The body will see this foreign protein, foreign virus, and then engulf it and develop antibodies to that by opening up this virus and killing the virus, opening the genetic material and processing it to produce immunity. So this is directly we are giving the virus here, but the virus here is in an inactive form, in the sense harmless vector is used to carry the coronavirus genes, and nothing of the infected material is allowed. So this is the one which penetrates the ACE2 inhibitory receptors and enters. And they are not given. You see the yellow one, nothing is there. The spike proteins are not given. So much so this cannot cause disease, but at the same time, it can elicit immune response. Under the cover here is an inactive, a non-infective, harmless vector is used. That is the technique. AstraZeneca Oxford vaccine, Gamaleya Sputnik vaccine, Johnson & Johnson vaccine are vector vaccines. The vector vaccines are two types, replicating type and non-replicating type. Replicating type means where we allow the virus to replicate and infect more cells and produce immunity, and the non-replicating is we are suppressing the virus not to replicate, so it doesn't replicate in the body. Only the genetic material of the virus inside, which is put in the wrapper, that is being extracted. So vector viral vaccines these are the two important vaccines, the Janssen vaccine and the AstraZeneca and Gamaleya Sputnik vaccine. Now, live attenuated vaccines. What are live attenuated vaccines? Here the coronavirus itself is taken and no viral vector is used, no mRNA is taken, no DNA is taken. The entire virus, coronavirus itself, is attenuated live. It is live virus, but attenuated means inability to develop or multiply or burrow into the ACE2 inhibitor. How do I do that? I do that by either by heat inactivation or by chemical attenuation or several lab mechanisms we can. For example, we are using similar type of vaccines for tuberculosis, chickenpox, polio. We are using live attenuated vaccines. Griffith University has developed SARS-CoV genome mutated, reduced to abolish the ability of the COVID-2 virus to replicate in the human cells. This is called codagenics or Indian Immunological Limited Biologicals also. These are also they are trying the live attenuated because this is very older technology but doesn't require very great sophisticated lab facilities. Unlike in the US and Western countries where the lab facilities are topmost, where you can extract the mRNA, you can do the DNA, you can do a viral vector. Here you are using a live attenuated. These are down the line in terms of their quality of scientific work, but then they produce a very strong immune response because we are giving all the proteins of the virus along with the genetic material. So much so the immune response will be quite good. At the same time, this cannot be used in immunocompromised, allergic people, people who are on immunosuppressive drugs, elderly people, people who are having drug interaction. These will not be able to be used. This is the restriction of this. Live attenuated vaccines advantages, live attenuated present advantages and disadvantages I have already told you. Now we will go to the inactivated vaccine. What is inactivated vaccine? The difference here is, here not only attenuated, the virus is dead. There is no more a live coronavirus. It is a dead coronavirus. Only the proteins of them, they go in, they cannot replicate, but only produce a host immune response. What is this? CoronaVac, Sinovac. These are the ones which are being done on this particular R&D line. In India also this CoronaVac is being developed. This is an older technology of using inactivated vaccines. The same information as we discussed earlier as far as the live attenuated goes here, immunocompromised, allergic people, people on immunosuppressive drugs, elderly people may not mount a proper response for these inactivated vaccines. So, this is the pros and cons of different vaccines. What did we learn? DNA-based vaccines, very good, but the problem is we are introducing the material into the nucleus. mRNA-based vaccines, we are introducing into the system, into the mRNA, into the cytoplasm. Not any viral protein, not the full virus, not the live virus, not the inactivated virus, but a small copy of the blueprint of how the spike protein looks like. And that also a Xerox copy. And that Xerox copy the body sees and produces the protein against the viral spike protein, and spike protein is inactivated and virus is not allowed to enter. That is the mRNA vaccine. Then adenoviral vector, the viral vector vaccines are, we can use a carrier virus. The carrier virus is a non-pathogenic virus. There are several viruses which are non-pathogenic to human beings. Some of them are adenoviruses. Pick up the adenovirus and put the coronavirus inside that and cover it up and give the package. Our macrophages will take the virus along with the covered virus, the coronavirus along with the covering, the sort of non-pathogenic virus, the adenovirus. They split open the adenovirus, recognize the protein and develop that. Two types here, multiplying type and non-multiplying type. Multiplying type, the body, the one viral particle you give, adenoviral particle, becomes 100, infects many cells there, cannot produce disease, but produces immunity. Non-replicating means you have one virus particle, one macrophage goes to the immune system and produces the antibodies. Then you have the what are called the live attenuated. Comparatively, the virus is live, but certain important proteins which are pathogenic proteins are removed. It is attenuated, means not allowed to do the pathogenic mechanisms, and not pathogenic, but only still it multiplies in the body and produces more antigens and then stimulates the antibody production. But sometimes it can mutate and can produce, disease, severe disease also in such a scenario. Inactivated, that problem is not there. None of the proteins are active, all are inactivated. All the viral proteins are inactivated, only genetic material is inside along with the virus, and then that is given. The advantages are, these are cheaper, but disadvantages are immunocompromised and that sort of group, whereas mRNA vaccines and the DNA vaccines and adenovector vaccines are little expensive. So viral subunits. So recently what they started doing is, why give the whole thing? Just take the subunits. After we are interested in the proteins which are on the outer surface of that, why don't I just take the protein alone without the genome? And without the genome, virus can do nothing. These proteins are injected in the system as subunits. They can develop immunity. Novavax and then chlorobiopharma is the S protein is directly injected. Now, VLP is virus-like particles. What here is the same virus, totally the same thing. There is no genetic material here. Genetic material is removed, it cannot multiply. Some of the spike proteins also removed. Remaining things only are given. This cannot multiply. But it is a virus-like particle. But still it can trigger the immunity. Not many vaccines are there under this category. Split viral vaccines. What they do, take the virus and cut it in the lab into 10 different parts and inject all the parts, so that it cannot multiply as a single unit, but the proteins are available for antibody formation. This is how the present day flu vaccine, Fluvarix you use, Fluvarix is a split viral vaccine. So likewise, there are examples of these things being used in our literature. Now, mRNA-induced antibodies. Here, this is not active immunity, passive immunity, where we take the mRNA and develop the antibodies in the lab, and this mRNA is translated and amplified to produce directly immunoglobulins, and these immunoglobulins are injected. These are mRNA-induced antibody vaccines, and this is a sort of novel way of introducing the passive immunity or antibodies. So this is all the story of different vaccines and their various types. Now, administration, DNA vaccines require a special delivery system because it has to go into the nucleus. And then 2 doses are used. RNA vaccines, syringe and needle are adequate, 2 doses. Viral vector, syringe and needle, 2 doses. Viral proteins, live attenuated, all through syringe and needle, only 1 dose they are saying. Inactivated, 2 doses. Viral subunits, 1 dose. So this may have some bearing because 1 dose is operationally easy to give, isn't it? If you give 1 dose and he is immunized, we are very happy. Community, people may not come back again for the second dose. There will be many people who will not turn up for the second dose. Those logistical problems are better off with 1 dose, but provided we have enough immunity generated by a single dose. Now, what are the risks of these things? Risks and responses and ease of production. DNA vaccine, risk of integration and mutation because the nucleus is touched. There is a sort of, the problem is the special facilities are required to produce those things. RNA vaccines are very safe and the immunity generated is very strong. DNA vaccines generate medium immunity, very strong immunity, very easy to produce. Viral vector vaccines are also risk of integration and mutation with viral vector because sometimes with the virus that is introduced which may not be pathogenic, with the mutation the virus may become pathogenic. But of course it induces very strong immunity and very easy to produce. When you use live attenuated vaccine, because it is live attenuated, risk of infection, it can mutate and produce infection. Very robust immune response because all the components of the virus we are giving, very easy to produce. But the problem is with the immunocompromised allergic that group. Inactivated viruses, Nothing can happen to the body, very safe, very strong immunity, but to generate large number, we require special facilities. We can't have millions and millions of doses because we require a large number of doses. These may not be the option, and the option may be somewhere between RNA or the viral vector. In the pipeline, several preclinical studies. After that, the investigation on new drug approval will be there. Then phase 1 clinical trials, then phase 2 and phase 3, then phase 4. What is the difference between phase 3 and phase 4? Phase 3 clinical trials are efficacy trials. Efficacy trials are under controlled conditions. You take two groups, one group you give vaccine, other group you placebo, double blind and controlled way you observe. Whereas phase 4, you release it to the community and see how the virus, how the vaccine is doing in the community in the effectiveness trial. And then Center for Diseases Control and Advisory Committee on Immunization, likewise we do have our ICMR and the drug regulatory authorities here. And then there will be 3 types of authorizations. One is called the compassionate use authorization, means, nothing is working, let us give, that sort of attitude. Emergency use, there is good evidence to say that it protects and use on the emergency use. And lastly, stockpile. That is, we have not authorized them, but then stockpiling is allowed so that in case the authorization comes, you don't have to wait for the stocks to develop. That is called the stockpiling authorization. So biomedical advanced research and development authorities are in place in every country, and they have to give the clearance. And we don't have to worry whether that is safe or not, because these people are expected to go very rigorously. These are not the pharma people. These are independent authorities, scientific community, who are looking at all these approvals, including the FDA, and the science should be taken care of. Then, what is the present state of vaccine? Oxford AstraZeneca, we know that it is a non-replicating vector type, viral vector type. Gamaleya Sputnik is also viral vector type, non-replicating, 2 doses, intramuscular, complete and complete. Moderna vaccine, same slide which I showed you earlier, which is again the lipid nanoparticle covering the messenger RNA. 2 doses, 0 and 28 days apart. Phase 3 trials are complete, being used in the United States. And Pfizer is being used in Europe and also in the United States. So there is a 3-layer LNP with mRNA, and again there is a 20 and 21 days. This also completed the phase 3 trial. Let us now compare the 2 vaccines that are available now. The mRNA vaccine that is being used, the study, Moderna vaccine, the study here, almost all the sort of ethnic groups are involved here, and the doses are 25 micrograms, 100 micrograms, and 250 micrograms. Or you see the quantity required to generate the immunity, very small quantity is required. We don't even require in milligrams. It's a very small quantity. Now you see the adverse effects. Everyone would be interested to know that any symptom, arthralgia, fatigue, fever, chills, headache, what are these? These tell us that the immunization is working, isn't it? Likewise, myalgia, nausea, local symptoms. Always, the severe is marked as red. That is seen in the higher dose. If you use 100 or 25, you don't see any severe. There were no adverse events, serious adverse events. There were no hospitalizations reported. So these are quite common with any immunizing agent. A couple of days of fever, myalgia, arthralgia, erythema, redness, induration is expected. Now, I told you here, the DNA stores the instructions for the protein. If it is a DNA vaccine, whatever you have given might be stored in the nucleus, and that might be passed on to various other cells that when they replicate. Whereas this is like a software mRNA. mRNA has a temporary set of instructions in the cytoplasm. And once the cell dies, until the cell is active, this is available for copy. Once that each cell has got a lifespan. When the cell dies, the mRNA also is inactivated, and it is not carried to the next group of cells, next divided cells. Not on next progeny, each cell divides, no, it doesn't get carried, whereas this gets carried. When you use a protein, this forms the basis of life, and this protein is made from the mRNA. From the DNA, this is a mRNA is produced, mRNA to protein. Our virus is a single-stranded RNA virus which is a positive sense virus. Positive sense virus means, suppose the coding is written A to Z, it is like that A to Z directly available. Negative sense means Z to A, it will be there. And then in the body, the mRNA has to reverse transcribe to get the correct picture. So negative stranded RNAs will have the code written in the upside down manner, and there will be another mechanism to make it into a mirror image is there, and that will be tilted to make a proper image, and that will be copied later on. Whereas this virus doesn't require that. It's already a straight copy is available. That's why the replication is quite faster with the COVID. It's a single-stranded, positive sense mRNA virus. mRNA is not RNA virus. mRNA virus is incorporated into the genome and multiplies. Whereas mRNA is the memory of that particular print in the cytoplasm against which the antibodies are produced. So this is the nucleus, whereas the mRNA are here in the cytoplasm, and this mRNA is engulfed by macrophages. Macrophages digest this cell and then present the mRNA to the immune site like the B cell, T cell, the memory B cell, memory T cell, and produce. And this mRNA has got these regions called the ORF, or open reading frame, in the center, and there is a 5' region and 3' region. This is to stabilize the mRNA. See that this doesn't disintegrate. Only the copying part required is only this one. Once it goes to our immunocyte ignores this and this because there are no instructions written here. Instructions are only here. This is the the white edge of that paper. It cuts off the white edge and takes only the lettered part and then takes the printing. These are called the ORFs. The open reading frame is only required. This virus has got several open reading frame. The one that is being used is the open reading frame related to the spike protein is used. Now, mRNA vaccines vis-a-vis comparison, BNT, Pfizer and Moderna vaccine here, phase 3 trials are also over. Total 43,661 subjects are recruited, out of which 41,135 have completed the phase 3 also. 30,000 people are recruited here. Total there were 170 cases of COVID occurred in this group, whereas 95 cases occurred in the interim analysis. This is still going on. This is completed. And out of them 170, 94% of the people are protected. Only 6% of the people in the vaccinated group developed as compared to the others. And regarding severity, there were 10 severe cases. 9 occurred in the placebo group and only 1 occurred in the treatment group or the vaccine group. How about here? Here there are 95 people who developed disease and 11 people developed severe disease. All severe cases are in the placebo group and the vaccine was 95% effective after the second dose. Now, these are the 2 vaccines which are tried against the disease. All other vaccines are trying against the antibody. How do I measure whether the virus is working or not? 1, it should prevent disease. It should generate antibody response. It should prevent asymptomatic infection. It should prevent reinfection. So these are the various parameters. Always trying to have 2 groups, group A with vaccine, group with placebo, expose them to coronavirus, see how many of them developed disease here, how many of them developed disease in the unvaccinated group. Nobody can do the trial, because it's unethical to expose the people deliberately to coronavirus. One group is vaccinated, other is unvaccinated. Both the groups are deliberately exposed to coronavirus. We can't do that. So they will be just like that in the community with all the protective measures, whatever are in place, and they will get the exposure to virus. And if the virus, the vaccine is protecting, the people in the vaccine group should be less affected compared to the people in the unvaccinated group. That is how the trial will be done. And also, none of these companies are having patients. These have registered several institutions to do the trials, and each has got around 20, 30, sort of international hospitals where the trials are being conducted. So no one single investigator has done that. Another thing is, the placebo and the information about vaccine is not known to the investigators nor to the company. It is a different monitoring group, international monitoring group, supported by the WHO, is the one which monitors the vaccine. If somebody develops a very serious side effect, that particular person's code is decoded, seen whether placebo or the vaccine, and then announced. If more serious events occur, the vaccine trial will be stopped midway. That's how these things are done. Not just like that, somebody cannot say, yeah, I want to promote the vaccine, I will just do it. It's not possible like that. Because one, there are no patients for the company. Number two, these are done in this is multi-centric trials. And the investigators are independent investigators, several countries. And three, the analysis and the data related to the status of vaccine and placebo is not with the investigator nor with the company. Now you see, Pfizer Biotech, you have to preserve at -94 for 6 months, it will be stable. But if you bring it out into 25 degrees Fahrenheit, which is around 5 degrees centigrade or around 0 degrees centigrade, 5 days it will withstand. Whereas Moderna vaccine, 25 degrees Fahrenheit or 0 degrees centigrade, 5 degrees centigrade, it is 1 month, it is stable. That is the basic difference in handling. This is stable only for 5 days, this is stable for 1 month. That could make a lot of difference in a country like India. And again, the duration, day 1 and day 22, and the antibodies are measured after 7 days. Here, day 1, day 29, antibodies are measured after 14 days. This is the first dose, this is the second dose, this is the antibody measurement. This is the first dose, second dose, antibody measurement. Here 95% effective, 94.5% effective. We have seen 30,000 people are included, different communities, different colors, 65 above and above patients, 7,000 are there, less than 65 with comorbidities, 5,000 people are there. There are total 95 cases, 90 in the placebo group, and then 5 in the vaccine group. 11 severe cases occurred, all of them occurred in the placebo group. Whereas here, there are 43,661 subjects have completed 171 cases, 70 cases, 95%, 95% effective. 9 of the 10 severe cases are in the placebo group. Only 1 serious case is in the vaccinated group. This is a vis-a-vis comparison of the 2 vaccines. So likewise, the CHAD-OX1 Oxford vaccine, which is typically called the Oxford vaccine, where we use a chimpanzee adenovirus as the vector. This is not pathogenic to human being, and we keep the viral material into this, the coronavirus. And 2 groups, one group is with the vaccine, the other group is meningococcal vaccine as the comparison group, and they have done that study. And you see, most of the side effects are mild or moderate. You can see from the graph, induration, itch, pain, redness, swelling, tenderness, warmth. And this is in the COVID, the active interventional group, adenovirus with the vaccine, and here with meningococcal vaccine. So more or less the side effect profile is same, and no severe side effects, no hospitalizations in either group. Now, you can see here, this is again the vaccine systemic reactions. That is, those are the symptoms and these are the reactions, more or less very mild and comparable. Now, this is the next one is AD5 vector vaccine. Here what they did was they used AD5 in one arm, the half dose. This is 5 into 10 to the power of 10. Here it is 1 into 10 to the power of 11. This is double the dose. This is 1 dose, full dose, and then the placebo. 3 arms are used. Dose titration also is done. Here also, as expected, the milder side effects are there more in the vaccinated group than in the placebo group, but none of the side effects are severe. And then you can see here, they measured not the disease occurrence, but looked at the antibodies development. This is an indirect way of saying that there will be protection. I don't know whether the antibody is protective or not. That question always remains. Whether these antibodies are neutralizing antibodies or non-neutralizing antibodies, we don't know. So this study doesn't include development of cases as the outcome, but the RBD binding antibodies. RBD is receptor binding domain antibody, that is the ACU2 binding antibodies, and other antibodies to SARS-CoV-2. These are all the neutralizing antibodies. are studied. This is neutralizing, this is the RBD antibody. And the vaccine group did much better compared to the placebo group. Now there is a nanoparticle vaccine here. There is one placebo arm and then one 25 microgram, twice, 2 times given, and 5 microgram followed by 50 microgram, 25 followed by 50 microgram, 25 followed by 50 and a placebo. Different combinations are tried in the NVX nanoparticle vaccine, and that also is under trial. Evaluating a vaccine candidate, we are interested how durable is the response. Durable means how long the protection lasts. Second thing is how safe it is. Third thing is the logistics like storage and distribution. These are the 3 areas. Time only will answer. One, the durability of immunity. See, what if it is durable for 1 year, we would be able to buy it, take it, because we know the flu vaccine every year we have to give. Whereas pneumococcal vaccine, we give once in a lifetime. Chickenpox vaccine, we give once in a lifetime. So some vaccines have to be given once in a lifetime. Some vaccines have to be given every year. Some vaccines we have to give a booster dose after 5 years. So if the durability is at least for 1 year, it is worth taking it. So that part we do not know because none of the vaccine trials have completed 1 year. So time only will answer. Whether it affects the asymptomatic illness, will it prevent asymptomatic illness? If so, it prevents transmission. How about the immunity issues in the elderly? In the elderly, we know the immune responses are not quite well developed. And how about the immune mounting response by these various vaccines? Is there a difference compared to the youngsters and the elderly in between different vaccines, between the doses? Long-term adverse effects of these we do not know. Short term, we have seen that they're like any other vaccine, and unless one waits for about a year or two, we don't know what will be the long-term adverse effect. Are the antibodies protective in the real world? So they have developed antibodies. Let us say these people who received the vaccine, are they protective antibodies or just bystander antibodies, which have got nothing to do with the protection? Sometimes are they harmful antibodies? That also is possible. In dengue fever, we know the one type of antibody, the non-neutralizing antibody, is a dangerous antibody. That's why second attack of dengue is dangerous. We don't know that. Acceptance by the public. With all these things, people may know, no, I am not going out. I am a lady. I already suffered, and I don't require. I am afraid. So many conditions they may say. The public acceptance also is a big thing. Government is actively promoting, but nobody can be compelled to be vaccinated because India doesn't compel anyone. Unlike in China, everybody is compelled to take the vaccine. Here it is not. Organizing mass vaccination may be an issue, particularly if you have 2 doses to be given, that too after an interval of 3 weeks or 4 weeks. Booster doses, we don't know when the booster is required, whether we require annual immunization, we don't know. These are all to be answered, only time will tell. Now, look at this diagram very carefully. There are people who look at the white part of it and the beautifully carved pillars. There are also people who look at the black part of it and try to find the black ghosts in that. In this whole world, we have people who are optimistic and who are pessimistic also. So it's always better to look at the rosy side of anything and be aware of what could go wrong also. Never be carried away by the first impressions of anything. And you see here, if you concentrate carefully on the white, you see beautiful pillars. If you concentrate on the black, pillars disappear in your eyes. Only you see people there inside that. So this is how we have to look at the current knowledge. And finally, the road appears quite clear, smooth, and there is sunlight ahead, but we have not yet reached the village where we are supposed to go. Still a lot of journey is there. We haven't reached the destination, but there is enough light to say that we can go. Road is not bouncy. Road is rather very well paved and very smooth, looks very nice. And don't forget the dividing line for Corona is SMS. What is SMS? S for sanitization of the hands, M for mask, S for social distancing. Social distancing, sanitization, and now the hands and then mask or inevitable dividing line between a rash driving on this lane and getting corona, or safely driving in this lane, the correct lane with the SMS, and not crossing the boundary and going ahead. Thank you all. A shot is not the panacea. Panacea means cure-all. It's not the panacea. Shot is not the panacea. We have to use shot along with the other available technology. Thank you all very much.